possibility of bringing any cells of the body into a state that resembles the original embryonal state. So I will cover this, and I will try, my hope is to leave you with an idea of what this discovery means for society as a whole, and also to give you an impression of what life in a lab is and some sort of ideas for your future careers. Is there anywhere particular where you would like me to focus? Is there any question in the beginning I can try and tailor my talk to? Yeah.
Maybe the thing we found out yesterday, the recent breakthroughs in stem cell research, you might want to include that because it's a more current breakthrough. The thing on the acids.
Yeah, okay, nature. Yeah, okay, so thank you. Another question? Okay, so I'll come back to this because it is a very similar approach to what I'll be describing, and it uses chemicals rather than scratching my head about my future, the way most of you will do very soon. And I've been working in very different environments. So I studied in Italy, so if you don't understand my accent again, interrupt. And then I had a very important 6 months in Sweden in '97, where I really went from being a medical student into experiencing what a lab meant. So for me, that was a very big breakthrough in terms of preferring the laboratory environment to the hospital environment, and this was very important for the rest of my career. And then in 2004, I went to start my life in the UK, and after a PhD I had in Italy with the Open University, so as a sponsoring establishment PhD, I did a postdoctoral training, and then I started having my independent projects, and now I lead a group in King's College that is focused on specific questions about stem cells. So if I can give you some suggestions from what I've learned in this particular path, it's not a very common path, but I think it's important to experience the different roles that sometimes make life back into university more focused to the real world. So I think one problem of a scientist is that sometimes it's very easy to kind of start living a bubble in your own world, and you lose track of it. But if you're considering even doing research, the option of doing medicine is still very viable, I think, because the course gives you an understanding of the body and an understanding of the disease, and it puts in perspective what these biological discoveries are. But at the same time, there is a passion and a rigor that you will only find in academic scientists. And it is a beautiful world, and it is a world that surprises you every day. But more and more, it's important to acknowledge that there are important figures that allow research to happen. So funding is always something you have to consider, and for example, there are very interesting models now where public and private funding comes together, focusing on one specific task that needs to be addressed. So just you can see that there are many different paths you can embark in if you want to specialize into a role. And you have to think about the cells in our own body doing a little bit the same. So the cells you have in your own body are very, very different to each other. And they perform very different functions. So you have to think of this a little bit as a career path. Do I want to become a brain cell, or do I want to become a lung cell, or a heart cell? And you can see immediately that they are very differently arranged in their patterns, and they're, even by imaging, you can see that they're very, very different type of cells. So a neuron will be able to irradiate axons and dendrites, so ramification to be able to sense other neurons. A cell in the lung needs to have elastic fibers in it so that it can kind of sustain the movement of the lungs. And cells, for example, like the ones in the heart, need to be able to form tubes and talk to each other in a specific way that allows the transmission of the electric signals and the contraction to occur. So it's clearly mind-boggling that all this diversity of cells originates from really one cell, contrary to what was thought in ancient times. So why are these original cells so special? What makes them so special? So it all involves understanding what stem cell means. And a stem cell is defined operationally as a cell that is capable to do two things. It is capable to replicate, but to replicate in a way that makes cells exactly identical to itself. And it's also able to specialize in a way that makes cells that can learn a function, like we have seen in the slides before. So these two paradigmatic patterns of choice between the self-renewal, so the capacity to generate cells that are exactly identical to yourself, or the differentiation, so the capacity to generate cells that learn a function, is the big dilemma that a stem cell is always juggling with. And as my mentor in Cambridge used to say, there is often a struggle between the cells and the scientist. A scientist would like the cells to go and do brain, but no, for some mysterious reason, the stem cell is still not decided what path to choose and what skills to be able to learn. And it's also very important to notice that the balance between the choice of replicating or specializing is very important to maintain health. So a disruption in this balance is detrimental for health. And typically, cancer is a disease of replication, so a cell gets a bit crazy and starts proliferating and is irresponsive to the signals that try to stop it. And on the other hand, diseases like neural diseases like Parkinson's or Alzheimer, where you start trembling or you start losing a bit of your head, depend from an imbalancement between the proliferation of the cells, the survival of the cells, and also the specialization of the cells that we call differentiation. But so far, what we saw is just a choice of whether to replicate and divide or learn a function. And it seems, up till a few years ago, it seemed like inevitable that a cell would progress once it embarked into a specific lineage into learning a function. So the big breakthrough that these guys came up with was managing to convince a cell that already had learned something to come back to a state where the choice would still be open. It's a little bit as if you were to become a medical doctor and then somebody took you back and when you're 10 years old and then decided that no, you have to become a lawyer, or I don't know, you have to drive a train or something like this. So it's really like some very, very difficult thing for a cell to try and forget all the skills that the cell has learned and come back to a state that is very, very similar to the original embryonal state. And in very simple terms, John Gurdon, the gentleman on the left, basically took a cell from a tadpole that already was specialized, brought it back into an egg, and generated a frog that was exactly having the same genetic information as the other tadpole. So basically, he came up with a frog from a cell. And in mammals, doing this is much more complicated. So without going into the details, Shinya Yamanaka did a very similar thing with mice. And now you can do very, very similar things with human cells as well. So in a nutshell, these two guys made a frog from a cell or a mouse from a cell. So how is this useful? Why is it so important? And why did they get the Nobel Prize? Well, if you could take from specific individuals their own cells, just from a biopsy, like from a piece of skin, and then brought them back into a stage where you could then redirect them into different functions, you could then theoretically inject these cells that have learned the function to cure the disease. As an example, if you have Parkinson and you're lacking some cells in specific areas of your brain, you can imagine you're going to pick up a piece of skin from these patients, bring them back in the laboratory to a state that is embryonal-like, and then reconvince them again to become exactly the same cell that you need for that specific function that stops you from trembling. So this is the very important breakthrough. And there's a consistent part of us working in regenerative medicine that is focusing on putting cells back into patients. There are nonetheless very important problems with this approach. Just to name two very immediate. If you take your own cells and put them back, organizing this becomes very, very problematic. It's very different from going to a pharmacy and buying an aspirin. Whereas if you take somebody else's cells and you grow them in big bulks, you can imagine that one day you could go to a pharmacy and get that specific cell type. But it's very difficult to have somebody else's cells in your body. So there's this big dilemma between, do I keep the doctors happy and I try use my cells, or do I keep the pharmacists happy and I try using somebody else's cells? So the whole cell therapy field is divided between this that is called autologous or allogeneic cell therapy. But this we're talking cells into is use stem cells in order to find drugs. And this I really believe it's a much more immediate and very useful area of research. So exactly with the same approach we can derive specific cells from patients, and then we can use the cells in a dish to understand better what is going wrong in that disease. Why are these specific cell types that make you trembling not present when I look in a plate, whereas if I take a normal person and go the same way, I would see them. And importantly, I'm going to try and give you some idea and flavor of my daily excitement using tools that several decades ago were not available at all. So as a common image of a scientist is the one that looks under the microscope. Now technology has reached us and has given us the chance to use very, very sophisticated analysis. So for example, now the microscope are typically capable to perform analysis in real time. So this is, for example, a simple device that lives in an incubator that keeps the cells happy at a specific temperature and takes images at a time defined by the user. And you can imagine that you can test drugs, for example, and then ask very, very specific questions like, which of these two cells is growing faster? Now you tell me. Which of the conditions do you think is growing faster? Yeah. Okay, so in this example, for example, adding a drug to the plate makes the cell fill up the plate much quicker than without the drug. And we don't know if they survive better, or they replicate better, or they are really truly the same type of cells, but we won't go into those details. Basically, you can screen a set of drugs and pick up something that makes the cells proliferate faster. And we know that some diseases depend on lack of proliferation. So you can think of picking up a drug that then could eventually be used for the people. And now things can get much smarter, for example. So this is an image that we generated in the lab, and it's an image that is based on two different types of cells. There's an underlying layer that you see in black and white, and then there's some cells that make colonies. But now the computer is capable simply on the morphology of the cells to distinguish between the two cell types, and you can quantitate, for example, the amount of colonies that are made, and you could still use the same approach with drugs. Even more sophisticated, you can then define better, so you can segment each single object within the plate, and then in the last four of these sets of images, you can ask, for example, what cell is switching on this light, which is a way we use to make sure that they are proliferating. So this is a computer-generated images that allows us to tell the condition of proliferation or specialization of the cells we have. And you can merge the two approaches. So now you can use videos of cells, but also you can use computer images to automatically make the computer understand whether a cell, like the red cells here, is embarking into the division. So with these tools, you can easily think that you have the possibility to compare the differences between one drug. And so, for example, once you have a very specific system set, you could really test in a plastic whether a drug is effective or not, way before this is put into the risky market where people could be benefit or not from this particular drug. And also, for example, in this image you have a drug that makes, once again, the cells proliferate, and you can see them, sorry, I keep saying proliferate, it's kind of division, so it makes the cell divide. And you can see that in the absence of the drug, the cells divide in the way they can do. So these are chromosomes that separate under the axe and they form two types of cells. But when the drug is present, the cells get stuck in the first phases of their divisions, and eventually they're going to die. So this, for example, is a work we have done to test some cancer drugs, potentially useful for brain tumors. And the hope is to try and get them stuck into the early phases of division, so that they then die and they don't reach the possibility of dividing. And in order to divide, a cell needs a machinery, like little organs like we have in our own body. And this structure, this beautiful structure you can see on the top right, is the set of organs that help the cells pushing away the two chromosome sets and allow it to divide. So by studying this more carefully, we can also understand the mechanism of action of some drugs that, for example, impair this process that is called the formation of the spindle in different ways, and they create beautiful images that are very different, and they can be instructive to understand what the drug really does. So just in a nutshell, trying to wrap up now, basically what I want to convince you is that we do have new tools that are available, and these tools can be used also to improve the drug discovery process, or to improve the possibility of finding very useful drugs. Now, I can relate to the question in the beginning. So basically, this process that is called induced pluripotency. So it is called a way to make the cell capable of generating different specialized cells. So pluripotency means that a cell can become different cells. It's typically done with very sophisticated DNA engineering. Whereas in the recent breakthrough, there are conditions that are very physical that allow this process to occur much faster. There is in recent times a very important shift in science between the use of small molecules and the use of DNA engineering, and now we are studying more and more how physical substrates are affecting the cells and how physical conditions are affecting the cells. So I hope this answers a little bit. And now, just to give you a final flavor of what I think is important, is that so there are different career paths to choose, but I think there is a very big misunderstanding about universities and academia. And this is that once you decide, for example, to go for a PhD, you're going to then stay in university and you're going to have a postdoctoral training, and then you're going to become a professor. And I think this is actually facilitated by university itself, but it's a big illusion. So these percentages that come from the report from the Royal Society in 2010, and things haven't changed so far, and they simply say that out of the bulk of PhD holders, there's a very big minority that manages to stay in a permanent long-term situation in university. And this is not by no means to discourage you into choosing a scientific career. It's just to open your eyes on the very different possibilities you have to do science. There is still a big separation between science that occurs in universities and science that occurs in industry, whereas I have been fortunate enough to experience the interface between the two worlds. And I promise, I think there is a big synergy between making something that is very rigorous and very smart and very intelligent, but also very, very focused on the real needs of a specific set of patients, for example. And just to describe a little bit more this difference, you can imagine that you are kind of trying to dig your path, and if you do it in university, whatever you find, wherever you go, if it's interesting and if it's new, will get credited and will get published and you'll become famous. Whereas in industry, you're given a specific task. So you're given a job which is to try and sort out a problem, try and find a drug for a specific disease. If 6 months down the line you don't want to work in Parkinson anymore, you want to work in Alzheimer, then you're out of industry. But in academia you will be still a happy person. So I think this may help kind of deciding in the future where to stand. But wherever you are, and by all means embracing the wealth of new tools available, I think it's very important to keep the values very clear and to stay very focused on what you want to achieve. So there is by all means a great necessity of passion. This is a job you cannot do if you're not passionate about biology and about how cells work and how cells decide to function. But there is a bit of ambition, which I think is good. But I hope I left you with the idea that staying very focused to the real world is also very important, and trying not to fly too high as a year. So for each cell we know what specific function it is going to have. So every fate of every cell is studied. For the human, not at all. So we, for example, have a complete black box in the intermediate, let's say, reservoir of cells. So we know a little bit about the N types. We know a little bit about the stem cells, but there are sort of transient cells we don't know much about. And these transient cells, for example, in the blood are a little bit more studied than in the rest of the tissues, and they are the ones that can then decide. But it's very similar to careers. So you can become a lawyer, but then you will become an international lawyer or a civil lawyer or a criminal lawyer. But you become a brain cell, and then you can become a neurons, or an astrocyte, which is a cell that supports it, or an oligodendrocyte, which is a cell that wraps the axon like a Swiss roll, but you're going to stay in the brain, if it makes sense.
I know you're in research and making drugs, do you only use human stem cells?
We personally do. So I work in the Human-Induced Pluripotent Stem Cell Initiative. It's a big project that brings about hundreds of normal and diseased human cells. But most research is, researchers kind of use every type of cells. There are a few organisms that are chosen. So Homo sapiens, the mouse, the worm I've just mentioned, and the fly, for example, the Drosophila fly, the little fly of the fruit fly, they are very well studied. So, for example, in some models of complex behaviors, and also the zebrafish, which is a very, very little fish, you can use one of these organisms even to model human diseases. So not necessarily you need to use human cells, but every system brings pros and cons.
You talked about how you could take a skin cell for Parkinson's disease,
Yeah.
but then surely if you've taken it from that person, isn't there the risk that it would then differentiate in the same way that caused the Parkinson's disease?
That's a very, very good question. Yeah, very good question. So there was a big hype maybe 10 years ago or so about gene therapy, right? So rather than putting back cells, in that case you would put up genes that kind of correct the typo that you have in the genetic code that generates that specific disease. So now a little bit of this field has shifted into the cell therapy bit. So you can imagine to take cells out, correct them in the lab, and then put them back so that they don't generate the disease anymore. Or you can do like we do, take them back and simply study. So compare it how in the plastic they don't perform the function, whereas the healthy cells do. And you can also intervene on the plastic. But yeah, it's a very good question. So also when you think of drugs that disturb this process of specialization, so that to sort of correct that, you need to keep into your head that maybe you can expand, for example, a pool of faulty cells, and you will always be finding the patients not benefiting from the drug. Yeah. Yeah.
I was wondering, when you take a specialized cell, is that you reverse it? so that you can get it to a state where it can specialize it. And what happens to the specialized features that it has? So like a nerve cell with a long axon coming off it. Does that does the cell lose that, or does it kind of absorb it back into it?
Yeah, another great question. So, yeah, in a nutshell, it loses it. So, you, irrespectively of the source, most induced pluripotent stem cells, so most cells that have been brought back to an embryonic-like state, are indistinguishable from embryonic cells. So they grow in those colonies like this one. They're very little, and they lose the morphology. And also, very interestingly, they also, if you look at the modification that occurs on their DNA, they really have the kind of like a blank slate of the marks that happens when you learn how to specialize. So when you kind of start putting out accents, your DNA is modified. Actually, not the DNA itself, but like the chromatin is the container of the DNA is modified. So that, let's say, DNA is like a Bible, or like a big book of like many chapters, and there's like different recipes. So in this book, there's also a recipe to do the branch, right? So if you decide to become a muscle, that chapter is kind of locked away. But if you decide to be a muscle, the chapter of actually fiber muscle is kind of scribbled, and the cell is working on that chapter. So when you bring it back to a non-specialized state, you already have the possibility to read most chapters of the book. Yeah, it makes sense. So there's a website here. You can see, I mean, for what King's College offer, there are a few possibilities, and there is a booklet here, and you can look into this. My career has been in the UK only in the last 10 years, so I can't help you with the specifics, but you can refer to this site if you need that. And I just leave you with very nice images of our group. So it's a very young, dynamic team, and this is what happens in most research groups. So I hope I haven't put you off to the potential careers, and I hope out of you there will be very bright scientists in a few years' time. Thank you.